So my name is George Warimwe, and I work at the KEMRI Wellcome Trust Unit in Kilifi, Kenya. And I'll be giving a talk on an introduction to clinical trials. And this is basically a lecture for people working in trial sites and anybody who's new to clinical research or wants to go into clinical trials. And the first place to start is really what is a clinical trial. And a clinical trial is basically a clinical research technique that is used for assessing the effectiveness of an intervention. And depending on the intervention, this may be a new drug or an existing drug, and you want to compare it with a pre-existing treatment, or what is the current treatment. It may also be for the purposes of preventing an illness, for example, a vaccine, and that's what falls under an intervention. As I mentioned, clinical research. Clinical trials falls under a broad term of broad class of research called clinical research, and this is just it's just research that involves human subjects. And some of the branches of clinical research include cohort studies, where you follow up 2 or more groups who have a disease or have been exposed to a treatment to observe a particular outcome. Another type of clinical research would be a case report, where you describe the history of a single case. The WHO defines a clinical trial as any research study that prospectively assigns human participants or groups of humans to one or more health-related interventions to evaluate the effects of health outcomes. And when we talk about health-related interventions, we're really talking about things that modify, health. For example, drugs, surgical procedures, dietary interventions. And the definition of health-related interventions by the International Committee of Medical Journal Editors just states that it's any intervention used to modify a biomedical or health-related outcome. How about health-related outcomes? What are they? So these include any biomedical or health-related measures obtained in patients or participants, including pharmacokinetic measures and adverse events. So these are things that you'll be measuring when you, give your intervention, be it a drug or a vaccine or a new technique. What is not a clinical trial are observational studies, and these are those in which the assignment of a particular medical intervention is not at the discretion of the investigator, and these are not clinical trials. Clinical trials involve giving investigational products, and these include drugs and vaccines, as I've mentioned. And what this is defined as an investigation product is defined as a pharmaceutical form of an active substance or a placebo being tested or used as a reference in a particular clinical trial. And these products may be there already in the market, but are used or formulated in a different way from the authorized form. It may also be something that may be a drug that's been used for example, cancer, and this drug is now going to be used for a different indication, and that's what an investigational product is. Clinical trials differ from standard of care basically because they are testing an intervention, and this is always in order to improve on standard of care. And you measure effects over a period of time, and you must have a comparison control group, and then it must have a method to measure. You must have a method to measure the intervention, and this is usually captured in the protocol, as you're going to see in a few minutes. And it also focuses on unknowns. For example, when you give a new intervention, you don't know what kind of effects that will have on the health of the individual. And these are things that are taken into consideration before medication becomes part of standard care. And that's why clinical trials are really important for working this out. So what are some of the basic concepts in clinical trials? There is Equipoise and Hypotheses, and this Equipoise is really testing whether there is genuine uncertainty or that one treatment or intervention is better than another. For example, if there's a drug A and a drug B, and you just have maybe anecdotal evidence that drug A works better than B, but you're not really sure, it's really uncertain, then that is what, in such a scenario, to really determine whether drug A or B is better, you need to perform a clinical trial to work out which of the two drugs is superior. Whereas in a hypothesis is where the investigator believes that there is a new intervention that is better than the current interventions that are being put in place, but there is no evidence to support his beliefs. And this is mainly, for example, with the studies on genomes and parasite biology, they often identify new putative targets of intervention, and these are what will be tested in a clinical trial to see whether, for example, if it's in a vaccine, where these targets are better targets for vaccines than pre-existing ones. So the next thing we're going to tackle is the protocol, and the protocol really is what establishes the question to be addressed by the trial, and it ideally just has one question, and this is the primary endpoint. One mistake that happens in clinical trials is when you have too many endpoints, and really the best design trials just stick to one primary endpoint, because you are more likely to answer the question and it's easier to achieve. Also in the protocol you may have some few secondary objectives, and these may be things that are related to the primary endpoint, but they should not distract from the primary endpoint, and some of the objectives may be exploratory. The trial is then designed around this, and as long as you stick to the protocol, you will most likely get your answer. There are several ways to design trials. Some, for example, are crossover trials, where all the participants in a given trial are exposed to the intervention in successive periods, and you then look at the behavior of these trial participants at the times when they were exposed to that particular trial. In factorial trials, for example, you have two or more interventions being evaluated separately, and in combination and against a control, and then you see how the different combinations, how that affects your outcome in relation to the control. You may have heard of trial phases, and there are basically 4 phases of clinical trials: phase 1, phase 2, phase 3, and phase 4. Phase 1 is the first stage of testing in humans, and it usually involves a group of 20 to 50 healthy volunteers, and the trials are designed to assess the safety and tolerability, and also the pharmacokinetics and pharmacodynamics of a drug, for example. In phase 2 trials, these are performed in larger groups of people, usually in the range of 20 to 300, and they are designed to determine the correct dosing and assess how well the drugs work, that is, efficacy of the drug or of the vaccine. In phase 3 trials, these are usually large multi-center trials, and they are randomized and controlled on large patient groups ranging from 300 to 3,000 or even more. And they're usually used to determine the treatment efficacy in comparison with what is currently the gold standard of treatment. In phase 4 trials, also known as post-marketing surveillance trials, this is where the safety of the particular intervention is assessed, and this is ongoing even after the drug or vaccine has passed through phase 3. And this is looking for very rare adverse events and any things that would be detrimental to the health of the participants. Something that needs to be done when you're planning a clinical trial, you need to know how well it will be suited for your particular environment, and this comes down to comparative effectiveness, and it simply defined as the conduct and synthesis of systematic research comparing different interventions and strategies to prevent, diagnose, treat, and monitor health. And you want to, when you're running a clinical trial, you need to know the circumstances of your patient. So, for example, if you're assessing an intervention that requires a certain level of technology, will the environment that you're doing this, will it be able to support that? So these are some of the things that you need to think about when you're planning a clinical trial. There is also pharmacovigilance, which is just pharmacological science that relates to assessing and understanding the prevention of adverse effects of treatments. This field involves monitoring, collecting, assessing, and evaluation of these adverse effects, and which will then inform whether something needs to be done for the trial, whether you need to correct or stop the trial in some cases. So it's a really important branch of clinical trials. Some examples of clinical trials involve some may be ward-based, for example, in a particular disease, for example, malaria. You may have very sick children who have malaria, and others are malnourished, others are having seizures, and you want to see if you give them a particular intervention, what happens to the, for example, if it's a drug, you want to see what are the pharmacokinetics in these different groups of patients relative to those that are not admitted, for example. Others may be community-based, which, for example, may include questionnaires and that kind of thing. And it may also involve like proof of concept trials, which happen in the community. So, have clinical trials improved healthcare? So, clinical trials date back to the time of what used to be called the trialists. And a good example is Dr. Lanes, who was a sheep surgeon and carried out a trial of oranges and limes for scurvy. This is in the mid-18th century. People like Paul Ehrlich and Alexander Fleming then came in later in the 20th century, and by 1950, the British Medical Research Council developed a systematic methodology for studying and evaluating different therapeutic interventions. Going back to how to design a clinical trial, the first thing we're going to talk about is a protocol. And a protocol is really what you see in a title of a lot of publications. It may be like a single center, placebo-controlled trial of maybe drug X when compared to Y. And in the protocol, you need to have details of who's conducting the trial, who's sponsoring it, where will it be conducted? Is it in Africa, in Kenya, whichever country is being conducted? And on whom will you be conducting the research? Is it a trial being carried out in children or in adults? And then you also need to put in the protocol what you are testing. Is it safe, and have tests been validated? Why is this research needed? That is, you need to justify why you need to do that clinical trial, and that's all in the protocol. And then what are the risks and what are the procedures that are going to be carried out on the trial participants? How will the data be collected? And how did you calculate how many patients you will need? These are all things that need to go into the protocol before you begin a trial. And basically, the protocol just states the question and explains how it will be answered. So you should have all these steps in the protocol. Some of the documents you will need together with the protocol are the informed consent form. As the title states, informed consent is just a form by which you gain consent from the trial participants to carry out the intervention. And it needs to be clear, and it needs to let the trial participants know what risks may be involved, what gains, and try and make sure that the trial participants know the difference between the research, which is the clinical research, and standard of care. And in many circumstances, for example, the local translation of clinical research or clinical trials may not mean what you want to put across. So, for example, in Swahili, research means an exploratory tests. This may be difficult to explain the difference between the standard of care and research in this case, and this is really a key principle when you're giving consent. You have to make sure that the trial participants understand what they are getting themselves into. And in special circumstances, especially in children and also emergency, you also need to think about how you're going to put this across, whether is it the parent who's going to give consent, is it a guardian, and when do you need a witness for this? Because then the whole point of GCP, which is what guides informed consent, this is good clinical practice. You're supposed to protect the rights of the subject, and informed consent, the informed consent form is supposed to be a means to attain this. The other documentation that you will need is a case record form, and this is what turns your protocol into a data capture system. And in this case record form, you list all the measurements, for example, that are listed in the protocol. And for example, if you will not use weight in your intervention, as a primary, as an endpoint in your trial, then it shouldn't be there. So you only put in what you're going to use for the analysis. And this differs from something called source data, which is where all the patient's notes and the lab reports are. And this needs to be available because the central concept of good clinical practice is that data is always verifiable. So the data on the case record form should match the data on the source data. The data on the source data, even if the case record form has just got the information that you need for the trial. Data is then taken from the case record form and entered into a database, which is then exported to a statistical package for analysis. Now, when you talk about database and statistics, you need to know how many numbers of patients you're going to use for your intervention. And these are things that you calculate using, with the help of a statistician. You need to know how big your study should be so as to detect the differences that you want. And usually, basically, the bigger the difference you expect, the less the numbers you will need, and vice versa. So this is really important. The protocol also needs to explain the objectives, the statistical objectives of your trial, but it is the report analysis plan that sets out how you will analyze the data. And you need to have this analysis plan before you begin the trial, just to avoid biasing the data. And then again, the database should be secure and should have an audit trail, which is currently difficult in non-commercial trials. So some of the keys to follow in the protocol, this is the case record form, the source data, and standard operating procedures. So you need the case record form, or CRFs as they're known. You should only collect data that is listed in the protocol and nothing else. Data taken from the case record form is then entered into the database and exported to a statistical package. And you need a standard operating procedure that lists all the operations of the trial, and translates the protocol into the practical and operational steps appropriate to your site. So who is involved in carrying out a trial? You have the investigators. These are the principal investigators. You have the coordinators and project managers, pharmacists, clinical officers, data managers, and entry clerks, and of course the laboratory staff, and also statisticians are really key for this process. So when you think about how a trial is started, you start by discussing an idea with your peers and colleagues, and this idea is then generated into a concept proposal. You prepare the budget and the consent forms and all that, as we have seen. You then take it through a protocol review meeting, open to all who are appropriate for that particular meeting. This is people who will be involved in the running of the trial. And then submit that final protocol or grant application if you're looking for funds to run the trial to the sponsor, who then go through the reviewing process once more, look at the ethics. This is normally done by the ethical review committee. And then you also need to engage the community and the data safety monitoring board members. These are the people who basically just monitor how the safety of the patients through the trial period, through the period in which you're running the clinical trial. And then you need to hire staff and write prepare the case report, case record forms as we've seen, the standard operating procedures. And once you have all this in plan, you'll be ready to begin your clinical trial. So there are international guidelines for conducting clinical trials, and this is all centered around good clinical practice. It's centered around protecting the rights of the patients, making sure that the patients are safe. And this comes from studies or experiences from the 1930s, where, for example, black American men were used in syphilis studies to study the cause of syphilis, and they were not treated. Following on from those earlier times, there followed an agreement between countries that there needed to be a global standard by which all trials are conducted, and this was to be in accordance with good clinical practice. And as I've mentioned, this protects the rights of those in a trial, but also those whose treatment will depend on the data. So you need to make sure that the intervention that you're testing is safe to the trial participants, but also safe for those who are going to use it, for those who will be treated using your intervention. So, and it essentially assures that the rights of the patients are protected by all those given a drug or intervention in the future based upon the data that you are collecting in the trial. So the definition of good clinical practice basically is a standard for the design, conduct, performance, monitoring, auditing, recording, analysis and reporting of clinical trials that provides assurance that the data and reported results are credible and accurate, and that the rights, integrity, and confidentiality of trial subjects are protected. Your clinical trial has to fulfill all these features of the ICH GCP guidelines. So good clinical practice basically involves collecting quality data. So this is data that is, so when you look at the case record form, your data should match what is in the source data. So it should be quality data, it should be verifiable, and it should have been collected in an ethical manner. So always thinking about the patient, and that is what makes up good clinical practice. Quality data basically involves data and that the data and the results that are reported are credible and they're accurate, that is, they can be verified. And then for the ethics, it's just making sure that the rights, the integrity and confidentiality of the trial participants are protected. So this is all covered in the ICH GCP guidelines, and your trial needs to run to fulfill these guidelines. So the basics on how to comply with GCP: you need to write a good and sensible protocol. That is, you need to weigh the risk versus benefits of the trial participants. You should keep your protocol simple. In this way, the trial should only be having one primary endpoint, because you're more likely to attain that objective or have an answer if you have one primary endpoint and follow the protocol to the letter. You also need to obtain ethical approval. You need to protect the subjects, that is by obtaining informed consent and ensuring their safety, their rights, and and confidentiality. You need to use a qualified study team. All the people who are involved in the clinical trial needs to be qualified. You need to handle investigational products appropriately. So, for example, if it's a vaccine, it needs to be kept in the right conditions. If it's at the fridge, it needs to be kept at a certain temperature, whether it's 4 degrees. And then you need to implement quality systems. So you need to make sure that, for example, as in the case of the vaccine, you need to make sure that there is a continuous monitoring of how the vaccines are kept, saying that basically the equipment that you're using for the clinical trial is up to date, is running well. And then you need to record and analyze the information appropriately. And you can't overemphasize the need to follow the protocol and trial standard operating procedures. And basically, it's just about conducting the trial ethically and being able to provide an answer that is reliable. And that is it. That is what you get out of when you comply with GCP. So on interpreting GCP, these are, as I said, are just guidelines, and they're intended to be interpreted in order to be appropriate for the relative risk of the trial. You need to think about how vulnerable the participants are, how risky the intervention is, whether it's a new drug or a drug that was, again, maybe used for a different indication, and how complicated is the protocol. These are things that you need to think about. A sensible application of a risk-benefit approach will help appropriate monitoring and meaningful consent and practical operation procedures. So other things to think about, you can ensure your clinical trial. You need to report any safety issues to the Data Safety Monitoring Board. You can register your clinical trial in a clinical trial register. You need to think about writing reports to your sponsor if you're being sponsored, for example, by a drug company. You need to think about how you're going to communicate your results, that is through publications, and the logistics of the daily running of the clinical trial, things like budgeting and transport. And as we mentioned, you need to think about storage of your intervention product, if it's a drug or a vaccine. And also data archiving. How are you going to archive the data? Is it going to be an open source, and what platform are you going to use for your data archives? And all the standard operating procedures, training records, and equipment service contracts, they all need to be filed in an easily accessible format. So finally, clinical trials are important because they are critical for improving health, because they evaluate new disease management methods or new drugs or vaccines. And there's no other way. It's the best way to evaluate a new drug or a new treatment, how effective a new intervention is. Taking part in clinical research can help facilities raise their standards as they introduce many useful tools and systems. More money than ever is now going into capacity building for clinical trials in resource-limited settings, because we want to improve health outcomes in these settings. And there are many opportunities for training and further qualifications, and really excellent career opportunities in clinical research. So and just a final note is that how we design and run trials is important for both the trial participants and for every patient or patients that might benefit from the data each trial generates. Therefore, it is an exciting privilege and a great responsibility to run these trials and to run them well in accordance with good clinical practice. Thank you.